Hello and welcome to my channel, Sam Biology Class. Friends, today we will discuss about restriction fragment length polymorphism. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Restriction fragment length polymorphism, RFLP, is a technique used to differentiate between individuals or organisms by analyzing their DNA sequence variation. It involves digesting their DNA with restriction enzyme and separating and visualizing the resulting DNA fragments based on their length. This technique involves gel electrophoresis, particularly agarose gel electrophoresis, southern blotting, and autoradiography. Restriction enzymes, also known as restriction endonucleases, are proteins that cut DNA at or near specific site known as recognition site. Each restriction enzyme recognizes a unique short DNA sequence, typically 4 to 8 nucleotides long. 4 to 8 nucleotides long. Restriction fragment length polymorphism also refers to the DNA fragments of different length that are generated after restriction digestion of different DNA samples by restriction enzymes. Steps of restriction fragment length polymorphism. First step is to extract the DNA from different samples. The DNA is then purified by a suitable method. Second step is restriction digestion. In this step, the DNA is digested with a suitable restriction enzyme. Restriction enzyme called as molecular scissors will cleave DNA internally at or near specific recognition site. This will result in the formation of DNA fragments of manageable size. The DNA fragments are then separated into distinct bands by agarose gel electrophoresis. Agarose gel electrophoresis is a technique that separates nucleic acids and proteins based on their size. After electrophoresis, the DNA fragments are transferred to a nitrocellulose filter paper by southern blotting. After transfer, the DNA bands or the DNA fragments located on the filter paper are hybridized with a suitable probe, which can be radio labeled or fluorescently labeled. Probes are short oligonucleotide chains. When incubated, the probe will bind to the DNA fragment which contains the complementary sequence. When the probe is radio labeled, the specific DNA fragment can be visualized by autoradiography. Last step is analysis. In this step, DNA fragments of different lengths obtained from different samples are compared and suitable conclusions are derived. These are two homologous chromosomes, out of which the first chromosome has 3 recognition sites for the restriction enzyme EcoR1. On the other hand, the second chromosome has only 2. This indicates that from this chromosome, the second recognition site is missing. Why? The answer is that it can be due to mutation. As mutation changes the sequence, the enzyme fails to recognize, because of which cleavage do not take place. In both of these chromosomes, this bold line represents the sequence which will bind to the probe during hybridization. So these fragments will be detected by autoradiography. When treated with echo R1, this chromosome will be cleaved at these three sites. As a result, two fragments will be produced. Out of two, only this fragment will be detected by autoradiography. This is because this fragment has the complementary sequence that can bind to the probe. So it will be detected by autoradiography. On the other hand, this fragment will not be detected because it cannot bind to the probe. Second chromosome, when incubated with echo R1, cleavage will take place at these two sites. As a result, only one fragment will be produced. This is because in the second homologous chromosome, the second recognition site is missing. In order to detect restriction fragment length polymorphism, both of these chromosomes are incubated with echo R1 separately. As a result, cleavage will take place at the recognition sites. Then the resulting fragments are separated by agarose gel electrophoresis into distinct bands. After separation, these bands are transferred to a nitrocellulose paper by southern blotting. In next step, the paper is hybridized with a probe which can be radio labeled or fluorescently labeled. After digestion of this chromosome, out of two fragments, only this fragment will bind to the probe. Hence, it will be detected as a single band after hybridization. In this homologous chromosome, cleavage will take place at two sites, and the resulting fragment, which contains the complementary sequence that will bind to the probe, will be detected by autoradiography. So there are two restriction fragments. One is smaller and other fragment is larger. Each fragment length is considered as an allele. So this fragment length will be designated as capital B, and this fragment length, which is larger than that fragment, will be designated as small b. This figure shows the allelic RFLP fragments of three individuals, and this one illustrates their inheritance. When both homologous chromosomes have three recognition sites, then only this band, which will bind to the probe, will be detected by hybridization. In case of other genotype where both the alleles are small b, that means when the fragments are larger, then also one band will be detected. But the DNA fragments present in this band will be larger than the fragment of that band. As a result, these fragments will move slowly through the gel. Because both alleles are same, so the DNA fragments will migrate as a single band. After hybridization, the single band can be detected on the nitrocellulose paper. In case of third genotype, in which one chromosome has three recognition sites and the other has two, in that case we will get two bands, because from one chromosome we are getting a small fragment and from other chromosome we are getting a larger fragment. So smaller and larger fragment will migrate at different rate. As a result, they will form separate bands which can be detected by hybridization. In addition to mutation, the size of a particular RFLP fragment can also vary due to the presence of variable number of tandem repeats V and TR between two recognition sites. In this example, we have two DNA samples, sample 1 and sample 2. For sample 1, these are the recognition sites, and this is the region that will bind to the probe during hybridization. In sample 1, BNTR has 5 repeats. On the other hand, for sample 2, these are the recognition sites at which restriction enzyme will cleave, and this is the region that will bind to the probe. In sample 2 DNA, VNTR contains only 2 repeats. Due to the presence of 5 number of repeats, when sample 1 is cleaved by the restriction enzyme, the resulting restriction fragment will be long. By contrast, due to the presence of only 2 repeats, when sample 2 DNA is cleaved by the same restriction enzyme, the resulting RFLP fragment will be short. This difference in the size of the particular RFLP fragment is due to the presence of 5 repeats in sample 1 DNA and due to the presence of only 2 repeats in sample 2 DNA. So in this way, presence of variable number of tandem repeats between the same recognition sites leads to polymorphism. Apart from this, other genetic processes such as insertion, deletion, translocation and inversions can also lead to polymorphism. RFLP has several applications. First one is genetic mapping, in which it is used to identify the locations of genes on chromosomes. Second application is disease analysis. This technique is also used to detect hereditary disease causing mutations. Apart from this, RFLP is also used in forensic science for DNA fingerprinting to identify individuals from DNA samples. In addition, this technique is also used in paternity testing to determine biological parentage and also to study genetic diversity within and between species. Friends, this is all about restriction fragment length polymorphism. For more updates, subscribe my YouTube channel, Sam Biology Class.